Yes, hi guys, this is our second video lesson. First one we did the quiz. This one we actually start to the new topic, which is, personally is really fun, ecology and the environment. So starting from the beginning, ecology is the scientific study of the interactions between organisms and their surroundings. It analyzes the interactions that living things have with each other and with their non-living physical environment. Okay, so if you can highlight this, which is the description of ecology, which is the study of interaction between living things and the non-living things in an area. Okay, carrying out, so an ecosystem is a distinct self-supporting system of organisms interacting with each other and with a physical environment. Okay, highlight up to this point. I will tell you when to highlight. Carrying on, an ecosystem can be small, such as a pond, or large, such as mangrove swamp or a large forest. This chapter looks at a variety of ecosystems and interactions that happen within them. So like I said, it can be it can be a pond in your garden, it can be a lake, it can be the ocean. Ecosystem is variable. Okay? And in the ecosystem you will have specific interactions between the organisms living there and the non-living factors. Non-living factors can be, for example, the sediment, the soil, the rocks, the water concentrations of minerals in the water, etc. Those kind of things. Components of ecosystems. So, whatever their size, ecosystems usually have the same components. Producers, consumers, decomposers. Producers are the plants which photosynthesize to produce food. Animals that are consumers, sorry, are the animals that eat the plants and other animals. Decomposers, organisms that break down that material and help recycle nutrients. So if you can highlight these, I'm not going to highlight the whole sentence. I'm just going to start to highlight with on the beginning of the sentence or the paragraph, and I'm going to put it in the end as well, because this way you will be able to read the actual sentence on the book, on the screen. And also, despite the living things, you will have a physical environment. Physical environment is all the non-biological components of an ecosystem. For example, the water, soil in a pond, or soil and air in a forest, like I mentioned to you. The living components of an ecosystem are called biotic components. The non-living physical components are the abiotic components. If you can highlight this as well, so living factors, animals and organisms, etc., plants. The biotic factors, and you have the abiotic factors, which are non-biotic, which essentially what it means, non-living things. An ecosystem contains a variety of habitats. Habitat is a place where an organism lives. Okay? For example, habitats in a pond ecosystem include open water, the mud at the bottom of the pond, and the surface of the water. Highlight these two. I'm saying it again, so whenever I put a highlighter on the beginning of the sentence and on the end, then you should highlight that whole paragraph or the sentence. So habitat is a place that an organism lives. An organism, for example, in a pond, it might live on the bottom in the mud like a worm, or it might live in the water column like a fish. Those are different habitats in an ecosystem. All the organisms of a particular species found in an ecosystem at a certain time form the population of that species. So this is actually the definition of population. All the organisms of a particular species found in an ecosystem at a certain time is your population when we talk about ecology. All the immature frogs, tadpoles swimming in a pond are a population of tadpoles. All the water lily plants growing in a pond make up a population of water lilies, etc. All the humans in our class make up the population of our class, for example. The population of all species, animals, plants and other organisms found in an ecosystem at a particular time form the community. So everything that's in there, every type of organism, not only one type, describes the community. So all the organisms that you found in that time at that pond is your community. Figure 14.3 illustrates the main components of a pond ecosystem. So here what do you have? The green ones are your abiotic factors, you see here. They are green: sunlight, air, water, soil. And you have your components of the biological community. So you have your biotic factors. You have your floating plants, you have the plankton, you have rooted plants, you have snails, tadpoles, etc., and other animals. Okay, is that clear? Hope that's clear. Carrying on. Oh, by the way, we forgot to read this. So decomposers are fungi and some species of bacteria. They feed by saprotrophic nutrition. You probably already know this. Saprotrophic nutrition means excreting digestive enzymes onto your food and then absorbing what's left, digested food. Carrying on, using quadrants to sample from a habitat. So quadrants, usually people mistakenly call them quadrants. A quadrant is quite different. It's a quarter of a circle. Okay, it's a different thing. But when we talk about ecology, we talk about quadrats. Okay? When an ecologist wants to know how many organisms there are in a particular habitat, it will not be possible for him to count them all. Okay, you cannot go in a forest and count every snail on there. It's hard. So instead, he is forced to count a small representative part of the population, called a sample. Sampling of plants or animals that do not move very much, for example, such as snails, can be done using a sampling square called quadrat. A quadrat is usually made from metal, wood, or plastic. The size of a quadrat you use depends on the size of the organisms being sampled. 1 second. Sorry about that. Yep. The problem with my microphone. The size of a quadrat you use depends on the size of the organisms being sampled. You cannot actually have a 1 meter square quadrant and you decide to sample pine trees in a forest. Okay, they're really huge, so you won't even fit one in a quadrant. So, for example, to count plants growing on a, let me move this out of the way. Count plants growing on a school field, you could use a quadrant with sides 0.5 or 1 meter in length. So 1 meter squared or 0.5 meters squared. As you can see in this picture on the right. By the way, let's highlight this up to here. Yes, it is important that sampling in an area is carried out at random. Okay, this is important to avoid bias. For example, if you are sampling from a school field, but for convenience you place your quadrants next to a path, this probably wouldn't give you a sample that was the representative of the whole field. It will be a biased sample. Biased means actually rigged. If you don't sample the whole area, you just sample, oh, there's mud here, I cannot go there, I will not sample there. It won't represent the whole population, the whole habitat, or the whole ecosystem. Okay, so it should be at random. And to make reliable random sampling techniques, we have different techniques with different strategies. So let's read the campaign. Some books and teachers talk about throwing quadrants. The idea is that you stand in the middle of a field and throw the quadrant over your shoulder. This is supposed to be random, but it isn't. The place where the quadrant falls will depend on where you stand, how hard you throw it, etc. It is wrong to use this method. Okay? Let's highlight this as well. So random, it doesn't actually mean just throwing it around and see wherever it lands, because it depends on who throws it. If I throw it, maybe it will go further. If you throw it, it will go nearer. So it changes from person to person, or where are you standing, or it might be even biased. You don't want to go all that far, so you don't throw it far away, etc. So here we have an activity that is actually shows us how to do a proper random sampling. Let's start reading. Practical using quadrants to compare the size of a plant population in two areas of a field. So if we didn't have this corona situation, we could have gone outside to do this, but instead we're going to learn about it on the book only. Sorry about that. I mean, it's not my fault. It's nobody's fault. But here we again. We'll stack at home. Anyway. One way that you can sample randomly is to place quadrants at coordinates on a numbered grid. Imagine that there are two areas of a school field, A and B. That seems to contain different numbers of dandelion plants. Dandelions are the yellow flowers. Maybe you have seen them. Yellow flowers that have, when they seed, the seeds, they fly away. They are like a puff ball. When you blow air into it, they just fly everywhere. Anyhow. Area A is more trampled, so it's more stepped on than the area B, and looks like it contains fewer dandelions. This might lead you to propose the hypothesis. Hypothesis, as you know, is your prediction for a reason, for a difference in two habitats. The dandelion population in area A is smaller than the dandelion population in area B. Why did we hypothesize this? Because the other one just looks like it. It's more stepped on. Okay, we know that it's more of a popular walking place, so to say. So we hypothesize this hypothesis before we do our experiment. In area A, two 10-meter square measures are arranged to form the sides of a square. Okay. A pair of random numbers is generated using the random number function on a calculator. Or you have different software that can generate random numbers to you. By the way, let's highlight these and carry on. So this is important. You actually divide your area into coordinates, okay? So for example, you have an area, you divide it into 1 meter squares, depending on how big is your quadrat. If it's 1 meter square, then you divide your area to 1 meter square coordinates, cells, so to say. Then on the left you have numbers, because it will be, for example, here you see it's 6. Coordinate is 6. Sorry, coordinate is 2 to 6, which will be the second meter of the horizontal plane and the sixth point of the vertical plane, which will give you this cell right here. And this number 2 and 6 is randomly generated using a calculator or a software. These numbers are used as coordinates to position the quadrant in the large square. Like I said, the numbers of dandelions in quadrat are counted. The process is then repeated for 9 more quadrants. So you generate 9 more numbers. You do small squares like that. Here, maybe here, 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 whatever. And these numbers, like this, are randomly generated. So it is actually random. It's not throwing it away randomly. It doesn't work, okay, like we discussed before. So this is actual proper random sampling method, we call it. The number of dandelions in the quadrant are counted. The process is then repeated for 9 more quadrants, like I said. The tape measures are then moved to an area B, and the process repeated to sample from 10 more quadrants in part of the field. The table shows a set of results for a study like this. So you did 10 quadrants, okay, for each area. And then you got some numbers. In quadrant 1 in area A, you have 4. In quadrant 1 in area B, you have 10. So then you calculate an average. Okay, calculate the mean number of dandelions per meter square in each area. Do the results support the hypothesis that the population numbers are different? How could you improve the reliability of the results? So if you take an average, basically 10 squares, you can basically add these numbers up. Okay? And you basically take an average. So let me have a calculator here. So you have 10 quadrants. Then we're gonna divide these numbers. We're gonna add them up, and we're gonna divide it by 10 for A. So plus 7 plus 1 plus 0 plus 3 plus 8 plus 3 plus 12 plus 1 plus 6. What do we have? 45. If you divide it by 10, average number of dandelions per quadrat is 4.5. 5.5 we don't actually use in studies like this, because you cannot have a half a plant, or you cannot have a half an human or an animal. So we just say 4 dandelions per square. So average number of dandelions per meter square, which is the size of our quadrant, is about 4. Let's do this for the other one, see if there's a difference. Remember, it's 4 for area A. So for area B, we have 10 plus 7 plus 9 plus 14 plus 12 plus 7 plus 16 plus 9 plus 11 plus 16. We have 111, which is a lot more than the area A. So let's divide this by 10. We will get 11.1, which is 11. So on average, in a quadrant, in a meter square area, you will have 11 dandelions in area B, but only 4 in area A. What is this? What does this tell you? Is you will basically, scientifically, you have more dandelions in area B. What was our hypothesis, if you remember, is that the dandelion population area is smaller than the dandelion population in area B. So which means we can actually, our hypothesis is true because we have more dandelions in area B as we calculated. Hope that makes sense. Sorry. Yes. Carrying on, let's see if we finish this. Let's carry on a little bit more, because I don't want to do a lot of pages in the first one to see feedback from you, and then maybe I can improve it. Or if you don't understand anything, we can just do like video lessons or video chats, basically similar setup like this. But let's carry on. The amount of variation shown by species in an ecosystem is called ecosystem's biodiversity. So how many different kind of species you have in an ecosystem? It's a combination of two measurements. So highlight this. The number of different species present, how many different species you have, known as a species richness, and the relative abundance of each species, their evenness of numbers, how many of each species you have. Okay. Take the example of two communities shown in table 14.1. So you have species. Community, as you remember, is all the species living in an ecosystem or in an habitat. You have species A, B, C, D, E. And community 1, you have number of individuals in each species. You have 10 individuals per each species. Okay, number of individuals in each species. In the second community, you have 1 from A, 1 from B, 1 from C, 1 from D, and 46 of E. So in here, you will have a lower species richness, because the population, the community is mostly consists of the individual, the species E. So let's read about it. Both communities contain the same number of species, 5 species, and organisms, 50 organisms in both communities. But community 2 is dominated by 1 species, okay, species E. Community 1 contains an even number of species, so it has a higher biodiversity. So the numbers of individuals are actually more evenly distributed. You have this is a better ecosystem. This is a healthier ecosystem, because this means nothing is being exploited. You have equal number of species, equal number of individuals per species. Each species have similar numbers, which means the biodiversity here is much higher. In the right one, the community 2, you only have a couple of numbers of the other species, but it is dominated by species E. To put this as an example, so invasive species come to in mind in communities like this. This might mean in species E is an invasive species. It basically harms all the other species, and eventually will actually kill them off. So you have a lot more of them in here. It's a less healthy ecosystem. It's actually a indicator of a less healthy ecosystem. This can be either maybe by pollution. Suppose this pollution affects all these species, but it doesn't affect this one. So there will be no competition for this species, so it will reproduce a lot. But these ones will eventually die. So again, saying community 1 is more healthy, more biodiversity rich community, the other one is less biodiversity rich. Carrying on. Alright, some ecosystems such as tropical rainforests have a very high biodiversity. Other ecosystems are dominated by one species. This is shown by the pine forest plantation of Northern Europe, which are dominated by one species of tree. The trees produce a very dense cover or canopy. Lack of light severely restricts the growth of other tree species on the ground layer plants. The pine forest also provides a very limited variety of habitats for animals. So like I said, but if you have a higher biodiversity, it's more valuable in a sense. Yeah, a tropical rainforest ecosystem contains thousands of different species of plants and animals, has a very high biodiversity. A pine forest has a low biodiversity. So actually, if you have a low biodiversity, your ecosystem is more, it's affected more when there's a change, which means all the organisms will die. But if you have high biodiversity, evolution is easier to take place. Anyway, reading about it on here as well, biodiversity is generally a good thing for an ecosystem. Ecosystems with a high biodiversity are often more stable than ones with a low biodiversity. This is because an ecosystem that is dominated by one or a few species is more likely to be affected by any sort of ecological disaster. So what I've been telling you about. For example, if a new disease arose that wiped out the dominated tree species, this would have an impact on other species that relied on the tree for food and shelter. In a more diverse ecosystem, other tree species might supply these sources. Exactly. So this is very important. Yes. Extension work here, I just like to mention it. It is possible to calculate a mathematical measure of biodiversity called diversity index. See, they're looking ahead, picture and other chapter. So biodiversity index is a number that you put these data in and then you calculate and biodiversity index. This is basically simple data we have. We can say, okay, this one has more biodiversity because it's obvious. But what if this was maybe 7, 5, 4, 7, 6, and this was 5, 5, 4, 5, 7? So how can you actually compare these two communities? That's when the species diversity index, the biodiversity diversity index comes into end. Okay. Let's actually finish this practical as well. Then we stop. Practically using quadrants to compare the biodiversity of plants in two habitats. The sampling method used in practical 17, the random sampling that we mentioned, can be modified to compare the biodiversity of plants in two habitats. Imagine that there are two other areas of the field, C and D, then simply contain different number of various plant species. A hypothesis about this observation might be the diversity of plants in area C is higher than in area D. 1 meter squared quadrants sampled are taken in each area. The numbers of each plant species present in each quadrant are counted. So as data may be similar like this, you will have at the end of the study. You will take 10 1 meter squared quadrants to each area. You use the same random sampling method. You basically create 10 by 10 section of area in each field. Then you divide it by coordinates and then you generate random numbers for these coordinates, and then you place randomly generated coordinated quadrants in this 10 by 10 meter area in each field. Then you calculate the number of species and number of individuals per species. So how many of individuals of a species you have. Then you will have something similar to this. Yeah, and then you have a table here. The table below shows a set of results. The student who carried out the investigation was only interested in comparing the broad-leaved plants into two areas and did not record grasses present. So basically sectioned off this data to only broad-leaved plants, which is clever if you're actually narrowing down your research. It helps you a lot, okay? But depends if this data is useful for what you're looking for. Of course, this is a limitation. Okay, I forgot to mention you. So, this is your hypothesis, and again, same study is used. The table below shows a set of results to carry that. Blah blah blah. We rate this. So, your plant species: dandelion, ripcord plant, broadleaf, plantain, daisy, and yarrow, creeping buttercup, white clover, common cat's ear, groundsel, etc. Then what you do, you have your number of species. So actually did not create the data by quadrants. This is actually total number of dandelions in all 10 quadrants. Sorry. Yes. My headset shut off. Carrying on. What was I saying? It's the total number of dandelions in all 10 quadrants, and you do the same for the area D, field B, field D, and field C. And exercise has plot the results as two bar charts, total number of UCBs and names. Use the same axis scales for different bar charts. Describe that biodiversity in two areas. Do the results support the hypothesis that two plant communities have a different biodiversity? So they actually have different biodiversities, because you can see this on data. You can see, for example, there's a big difference here. The bar chart of this will be more skewed on the middle. Okay, you have majority of these two species, but here it's more more diverse. You have a varied number of species, varied number of individuals of different species. But here you have a lot of these two species, and then the other ones are much less. So they have a different biodiversity. As you see, you can actually make assumptions very easy, because you need to either see it on a graph, or you need the species diversity index. So if you use that formula and put numbers in and calculate it, it will give us a number of biodiversity. So if area C has a higher biodiversity, it will have a high species index number, and so on. It will have area D will have a lower one. Well, anyhow, so I will stop it right here. Hope it went well for you. But like I said, I'm looking for your feedback. If you have questions, just ask me, and I will send you a video list that you should watch about ecology, which I think is pretty helpful and nice. It might be quite fun as well. I don't know. And thanks for listening, guys, and talk to you again.